Thank you for the kind introduction, and thank you for the invitation, CFRI organizers. Unlike you experienced earlier speaker, his name is Sriram, I'm very slow talker, so if it's too slow, just let me know, speed up. So the title is slightly revised, Improving CF Airway Mucociliary Clearance by a combined beta adrenergic and cholinergic agonist. Before I start, I acknowledge first because we tandemly speaking. Also, a lot of people I need to acknowledge, but especially Jeffrey Wine, Carlos Milla, and Jack Sellers at Stanford, as well as John Engelhardt, who provide precious CF animal tissues to us, as well as collaborating Canada, Xiaojian Luan and Juan Janowski, and all those CFRI, CFF, as well as Stanford IMA for the financial support. As a background, mucociliary clearance is vital airway innate defense mechanism, and it is impaired in muco-obstructive disorder, including CF subject, and contribute to lung disease. And CFTR modulators, as you heard multiple times at this meeting, improve mucus clearance in most CF patients. However, people whose mutations are not targeted by CFTR modulator, like 10 to 30%, depending on the ethnic group, remain at high risk. So increased mucociliary clearance could help such a patient. So the question was whether the impairment in mucociliary clearance found in CF patient is primary or secondary to inflammation infection. So the answer came from animal study, newborn CF pig and newborn CF ferret, as well as young CF rats. From those studies, scientists discovered that mucociliary clearance is a primary defect. So MCC impairment precedes infection in these animals. That means human, the impairment of mucociliary clearance is primary. So to do mucociliary clearance experiment in ex vivo trachea, our colleague Jinyak Chung developed this simple but eloquent method to measure mucociliary clearance velocity. Briefly, you can dissect the whole trachea from either newborn piglet or ferret trachea and cut the whole tracheal tube and open it through a posterior smooth muscle layer, and then pin it down in a way apical side is toward to surface to the air, 95% highly humidifying air condition, and basolateral side contact to the bath, as you can see inside the view of the mucociliary clearance setup. And the tissue is like mounted in a boat to prevent the entry of bath solution, as you can see in the top of the panel, pig trachea. And then you pin it down. And when you drop the Xerox particles, you can see unidirectional movement of mucociliary clearance. And these are all being recorded with a time-lapse digital camera. While we are learning various drugs, including cyclic AMP agonist and the calcium agonist Carbachol in different doses, we found that if you use forskolin, like 10 micromolar, typical concentration, 10 micromolar, you can increase, this is a time course of MCCV, mucociliary clearance velocity. As you can see, forskolin alone increase in here. Well, you can see in blue circle, that is forskolin induced mucociliary clearance velocity. And the red square is MCCV induced by low dose of Carbachol. So they produce like that MCCV. If you combine those two, that is additive effect. However, if you combine those two drugs and then treated it it is much larger than additive effect. So it's a synergistic effect of mucociliary clearance velocity by those two combined agonists. So the hypothesis to be tested was low-dose cholinergic plus beta-adrenergic agonist accelerate mucociliary clearance velocity more than their predicted additive effects, so synergistic. Well, if you think about the concept is counterintuitive because response to beta-adrenergic cyclic AMP agonist is missing or defective in patients with CF. And cholinergic calcium agonist is known to induce airway constriction. So it's not good, it's dangerous. So that is why the therapeutic possibilities of combined agonists might have escaped notice previously until we discovered. So importantly, this can be produced, the synergistic mucociliary clearance velocity can be reproduced in CF airway as well. This is CF ferret, again, time course of mucociliary clearance velocity is showing here. As you can expect it, forced choline alone, that is blue circle, and there is almost no increase of MCCV, but when you add carbachol alone, it induces somewhat. And then when you combine those two, as you can see, it produces again synergistic mucociliary clearance in CF ferret. If you combine those data in a box and whisker plot, you can see the synergy response to the right, the yellow one, the box is significantly larger than arithmetic sum of mucociliary clearance velocity induced by forced choline or carbachol. So we tested it in piglet and it works fine with the forced choline and carbachol. And more importantly in here, a clinically approved inhalable drug, formoterol and methacholine also work as the other drug like forced choline and carbachol. As you can see here, forced choline, formoterol alone increase some mucociliary clearance velocity as well as methacholine. But if you combine those two, as you can see, they generate synergistic mucociliary clearance as well, showing in the box plot too. As I mentioned briefly, the cholinergic drug is causing airway constriction, so it's dangerous. So we need to know whether beta agonist can prevent cholinergic drug airway constriction. So we developed simple method by slicing newborn piglet or ferret trachea, like a 1.5 millimeter thick. And then in the presence or absence of beta-adrenergic plus cholinergic drug, you can see whether the lumen area is changed. By measuring it, we can assess whether there is airway narrowing or not. So as you can see there, by cholinergic drug alone, it can reduce the lumen area, while in the presence of beta agonist, it can prevent the cholinergic drug-induced airway constriction. So we tested in both ferret, wild-type ferret, CF ferret, as well as piglet, and it all works in the presence of pretreatment of beta agonist first. But in this case, interestingly, as you can see, when you run the experiment, first baseline and then combine those agonists, synergy agonists simultaneously, it induced, as you expected, mucociliary clearance velocity, synergistic MCCV, but it also can block cholinergic drug alone. As you can see, the red square, that is methacholine alone, that causes airway narrowing, but when you add simultaneously, it still can block methacholine-induced airway narrowing. So it's safe in this way to using simultaneously. Potential contributor to synergistic mucociliary clearance can be MCC reflects transportability of mucus and ciliary bead frequency, and transportability is largely controlled by the volume. For example, airway surface liquid, ASL, and the pH of the mucus. And ASL height and composition are determined by fluid mucus secretion by submucosal gland and surface epithelial, and fluid absorption by surface epithelial cells. So first we test whether synergy agonist can induce synergistic glandular secretion. This is one way of doing it in situ single gland secretion experiment. This sample is pink mucosa dissected from underlying cartilages and mounted in pliable silica petri dish, and surface is cleaned and dry and add water saturated mineral oil, as you can see in the center. And you can assess drug in basolateral side, whether it can generate the bubble. You can see the small bubble because of the condition here. You may not be able to see well. But anyway, the movie is 20 minutes of baseline secretion followed by 30 minutes of low dose carbachol cholinergic stimulation followed by 30 minutes of synergy agonist. Take a look. This beta and carbachol alone. And now when you add both drug, you can see a lot of glandular secretion came out from the gland. So if you plot it in wild type pig, wild type ferret, as well as CFTR knockout ferret, you can see all synergy response is significantly larger than the arithmetic sum of secretion rate by forskolin plus carbachol, as you can see, all these three samples. And while previously, while we study proteomic study with the individually collected glandular secretion in collaboration with John Engelhardt using LC and tandem mass spec, what we found was one of highly secreted protein from the gland is that one. It's called BPI fold protein. It's called like a bactericidal permeability increasing proteins. Those are well known. It's antimicrobial activity. And the proteins are also called as a plunk, PLUNC. And there is a two type of plunk. One is long form and the other one is short form. So short form of S plunk is well known for its potent anti-proteolytic properties. So that way it can suppress inactivity. What they do is they inhibit inactivating proteases. So indirectly they suppress inactivity as well. So keep in mind the mucus that secretes from glands contain lots of ENaC modulating proteins. So this is one of them. Take a look. And this is again a time course of mucociliary clearance velocity plot using a newborn piglet trachea. So one is the green triangle is pre-treated with the benzamyl. Benzamyl is sodium channel blocker. We use 10 micromolar of benzamyl in the previous like 30 minutes. And then with and without benzamyl, you can follow the baseline mucociliary clearance velocity and then add synergy agonist. When you look at the baseline MCCV, benzamyl pre-treated trachea shows like 6 times significantly higher MCCV compared to not treated one. But when you combine those two drugs, synergy agonist, there is no difference, suggesting synergy agonists already suppress inactivity. In part, they secrete synergistic mucociliary glandular secretion. So those mucus can, when they come out, they can suppress inactivity on the surface. And this is inconsistent with our earlier report with short circuit current measurement in pig tracheal mucosa, you heard from other speakers like Jack Sellers and other people saying about the short circuit current. This is a similar thing. By adding forskolin, you can see the short circuit current traces going up. That means it's like anion secretion is increased, chloride and bicarbonate secretion is increased. And then when you add the low dose of carbachol, 0.3 micromolar of carbachol, it produces transient short circuit current peak followed by sustained short circuit current decrease. After those decrease, when you add INEC blocker inhibitor, benzamide, you have only small piece of decreasement. That means this synergy agonist already suppress INEC activity. So in collaboration with Jack Seller's lab, we also measure bicarbonate secretion by synergy agonist. As you can see, comparing to basal bicarbonate output, the rate of bicarbonate output is significantly increased by formoterol plus methacholine. So far, the data shows that synergy agonist induce the synergistic airway glandular secretion and increase and suppress ion fluid support transporter, for example, chloride bicarbonate secretion and sodium absorption, respectively, across airway surface epithelium. So then the question is, do synergy agonists can increase ASL height? So in collaboration with our colleague in Canada, Janowski group, we measure ASL height using synchrotron X-ray beams in phase contrast imaging. As you can see, here is time course of delta ASL height, a micrometer, expressed in the micrometer. And the first one is the black square is no agonist at all, like the pig trachea has no agonist, and this is a control and DMSO. And when you add the formoterol alone, it increases ASL height, as you can see in the red square. And if you combine formoterol plus methacholine synergy agonist, it further increases ASL height. So when you do the slope is like a rate. And if you plot in different way, synergy agonists significantly increase the rate of ASL height in pig. So this is a proposed mechanism. If you think about those small area of trachea, and the top portion is now airway lumen inside of the tube, and the yellow portion here is well, this is a mucosal gland and on the top, and then here is surface epithelial cells, and this is the ASL height expressed as a yellow line. And as you can see, during the baseline period, L rest, some mucosal gland as well as surface epithelial cell secrete chloride bicarbonate as well as fluid that is balanced with the sodium absorption and the fluid absorption by through INEC. But when there is a beta agonist around, a beta adrenergic agonist around, then it predominantly secrete chloride bicarbonate through CFTR and fluid as well. And if there is a cholinergic agonist, then it chloride bicarbonate can secrete through calcium activated chloride channel, as well as carbachol can suppress INEC activity. So if you combine those two synergy agonists, ASL depth is increased much more. So MCCD is at basal and increased. If you combine those two, they are synergistically increased without airway narrowing. So the summary is beta adrenergic plus a low dose of cholinergic agonist synergistically increase mucociliary clearance without airway narrowing in ferret, pigs, and most importantly in CF ferrets. Our data suggests that synergistically increased mucociliary secretion, submucosal gland secretion, decreased ENaC-dependent absorption, and increased bicarbonate secretion and ASL height are components of synergistic increase of MCCB. The result of more exciting result of in vivo sheep MCC study, the effect of synergy agonist on CF cells plus minus CFTR modulators and partial outcome of phase 1 safety trial in human will be followed by our colleague, Dr. Carlos Milla. Thank you.